Hello and welcome to another informative webinar from Diagnostic Solutions Laboratory. Today's webinar, SIBO Challenges, Key Insights with GI Map, is presented by Tom Fabian. Tom is a clinical education consultant and science advisor for Diagnostic Solutions Laboratory. He is a former biomedical scientist with deep expertise in the role of the human microbiome in health, exercise, chronic disease, and aging. So whenever you're ready, Tom.
All right, well, thanks so much, Ali. I appreciate it. And I hope everyone is having a great day today. So what we're going to be focusing on today is the topic of SIBO and how it relates to GI map. So of course, GI map is not a SIBO test. However, it can play a pretty big role in helping to resolve a number of challenges that clinicians typically face when they're trying to diagnose and also treat SIBO. So this next slide is a list of some of the most common challenges that clinicians tend to encounter in clinical practice when they're dealing with SIBO. So there's really kind of three main scenarios that we tend to see when clinicians opt for also including GI map as a way to assess some of the challenges with SIBO. So these scenarios really break down to three things. The first one is pretty much when clinicians suspect SIBO based on symptoms, and so they go ahead and run a breath test for their patients, and it turns out that that breath test is negative. So that can certainly present challenges because that's sort of the initial suspicion based on symptoms, and yet that begs the question of what are the next steps? What do we need to find out in terms of what's accounting for those symptoms? The second most common scenario tends to be when patients are positive for breath testing. So they're positive for SIBO based on the diagnostic criteria, and then clinicians go ahead and treat accordingly. But then, unfortunately, in many of these cases, they only find that patients are responding partially with treatment. So they sometimes will respond initially and then relapse, or they may not really get much symptom resolution. So of course, that begs the question of what's going on there, and does that warrant retreatment? Does that warrant taking another approach? Or kind of going back to the drawing board and looking to see what else may be going on in the gut. And then the third common scenario is very similar. So when patients are positive for breath tests, so SIBO has been confirmed based on the test, oftentimes even with multiple rounds of treatment, many patients experience a recurrence of SIBO symptoms. And then if SIBO testing is repeated, sometimes they also see that breath test results are also showing that they're continuing to be positive. So this can be quite a challenge, and we do see this pretty frequently in patients that have been suspected or diagnosed with SIBO, and then the clinician decides to run a GI map to see what's going on. So this is really a big area because we know SIBO is something that's commonly assessed for a wide range of patients, partly because those symptoms tend to be so common. So just kind of as a recap, most of us are already really well aware of this, but the primary symptoms of SIBO, at least the most common ones, tend to be bloating, abdominal bloating and distension, excessive gas production, abdominal discomfort or pain, and then diarrhea typically, or constipation, or some sort of mix of the two. So those again are far and away the most common symptoms, often attributed to SIBO. And as we'll get into a little bit later on, as you'll see as we go through various cases, that of course there can be many other potential causes or contributors to these symptoms. So of course that's a challenge for clinicians to decide what is it that's really attributing or causing those symptoms. So I want to dive into a number of cases here to start off with. So we'll cover 4 cases in this webinar, and then working in some of the evidence in these different scenarios in terms of the research that's out there that helps us better understand what may be going on in these cases. So this first case is a patient that was suspected of having SIBO based on symptoms. The clinician ran a breath test and the patient was negative. And so after the breath test, patient was still experiencing lots of bloating, in this case quite significant, along with alternating constipation and diarrhea, so IBS-like symptoms. In addition, this patient was experiencing long-term food sensitivities, as well as issues with acne, fatigue, and weight gain. And then a side note, also this patient had a pretty high level of stress as well, that of course, we all know that that can also impact GI symptoms and particularly digestion as well. So looking at the GI map results, first page was clear, no pathogens were detected. Second page, no parasites or viral pathogens were detected. H. pylori was also negative. But when we look at the normal bacteria flora section, we start to see that there are some imbalances. In this case, a mix of some that are high and some that are low. So that's a fairly common pattern that we see where there's a bit of an overgrowth of some types of bacteria, as well as a deficiency in others. Now, we've covered the key dysbiosis patterns that we often see with GI map in other webinars. So I'd encourage you to check those out to learn more about those types of patterns. But in this case, we're seeing some evidence for a mix of insufficiency dysbiosis, which is basically a lack of some of the key beneficial bacteria, and then also a little bit of an overgrowth pattern, which is something we commonly see when patients have digestive dysfunction type issues, SIBO type symptoms, et cetera. We also tend to see an overgrowth pattern in this opportunistic bacteria section. As we're going through, I'm calling out some of the markers that are potentially most clinically relevant for these given cases. So in this case, in the opportunistic bacteria section, we can see that Bacillus was high, Pseudomonas was high, and then also Streptococcus. And I'll be talking about particularly Pseudomonas and Strep as we proceed through these cases. But in this case, Pseudomonas seems to be one of the more notable ones that could be contributing to symptoms based on what we know about Pseudomonas and also what we see clinically. Autoimmune triggers was negative. The two here that are detected, Prevotella and Fusobacterium, are common and detected in virtually everyone. So those two are really not notable unless they're officially considered high. In the fungi yeast section, we can see in this particular case, Candida was also high. So now we have two fairly significant opportunistic organisms that we see that are elevated in this patient that was suspected of having SIBO, but the breath test was negative. Parasite section was also negative. Intestinal health section, nothing was officially flagged as high or low. So on the surface, things look pretty good. But definitely, we encourage you to scrutinize the numbers because some of these may be close to the high or low limits. So they may essentially be suboptimal, or they may be close to a range that would be considered sort of high normal that can still be clinically significant. And that's certainly true of this marker that we flagged here, the anti-gliadin IgA. So you can see that the cutoff for being considered high is 157. And the anti-gliadin result for this patient was 152, so quite close to that cutoff level. So certainly we would consider that suspect and probably high normal. So that may be indicative of some gluten reactivity, for example, or just a general set of circumstances that are leading to a higher level of reactivity overall. So the case summary here is in terms of the most clinically significant findings from this report that are related potentially to the patient's symptoms would be high Candida and high Pseudomonas, some general overgrowth, as well as the elevated anti-gliadin that's almost at the level we would consider high. So I just want to dive in a little bit about some of the background information, particularly on Pseudomonas. So this is a topic that we have addressed to some extent in other webinars and resources, but just want to reiterate here because it does seem to be something that's quite clinically significant in general, particularly based on a lot of new research that's been coming out in recent years. But it also tends to be one of the organisms that we commonly see elevated in patients that either were suspected of having SIBO and then tested negative based on breath testing, or patients were positive for SIBO but did not respond well to treatment. So again, it does seem to be quite clinically relevant in many of those cases. So there's a couple of studies that have been conducted over the last few years that have linked overgrowth of Pseudomonas to irritable bowel syndrome type symptoms. So that's often related to SIBO as well. And they summarize the finding here where they say this study shows that Pseudomonas aeruginosa is detected more frequently and at higher levels in IBS patients than in healthy subjects, suggesting its potential role in the pathophysiology of IBS. So at that time, not as much was known, of course. This was published in 2011. Since then, a number of studies have been published to give us more insights into some of the molecular mechanisms for how Pseudomonas might be contributing to symptoms in patients. And this was a major research study that was published last year, 2019. And again, we have featured this one in a couple other webinars because it does seem to be related to a number of different scenarios, including food sensitivities as well. So in this article, they say we have recently shown that undigested gluten can be metabolized by certain opportunistic pathogens, such as Pseudomonas aeruginosa, leading to the production of peptides that more easily translocate across the mucosal barrier with retained immunogenicity. So in this case, they're following up on earlier research that has linked Pseudomonas to gluten sensitivity and possibly to celiac as well, at least in terms of worsening symptoms. And it appears to do that by the production of a particular enzyme that it releases, and that's a protease enzyme, so it breaks down proteins. And it actually can also break down partially gluten proteins into smaller fragments that can then more easily cross the intestinal lining and then are more likely to stimulate the immune system. And now a lot more research has been done on this particular protease that it produces, and it's considered a virulence factor. And it actually plays a wide range of roles in the negative effects of Pseudomonas, including promoting biofilm formation, and in particular promoting inflammation directly, and that's been shown mostly to happen so far in the duodenum, so the upper GI tract. So it's relevant, of course, to the small intestine and small intestine pathology, and possibly resulting dysbiosis from that inflammation. So again, it's something that's been shown in a number of studies, but definitely appears to often contribute to inflammation, particularly in the upper GI tract, where it may have many different effects. In this study, they mostly linked those effects to an increase in the risk for gluten sensitivity and also the broader picture of food sensitivities. So now I'm going to go into a second case example here. In this case, the patient also was suspected of having SIBO. Also, in this case, the breath test was negative. So SIBO was not confirmed, but again, the symptoms of SIBO were present. In this case, the clinician tried a low FODMAP diet just to see if that was going to help. It did seem to help just a little bit, but not much. So really only a little bit of symptom relief from the low FODMAP diet. This patient also had established food sensitivities as well as weight gain. So of course, food sensitivities may be part of the symptom picture as well. We look at the results of their GI map test. Pathogen section was clear. Parasitic pathogens, viral pathogens, and H. pylori once again were negative. In this case, we also see a bit of a mixed pattern under the normal bacteria flora section, with one of the bacteria groups, Enterobacter, just a little bit elevated. In this case, Akkermansia was low, below detection limit. And then Bacteroidetes was also low. So a little bit of a deficiency pattern. Akkermansia and Bacteroidetes tend to be more indicative of what's going on in the colon. So they're colon resident bacteria by and large. So that appears to be part of this picture here is a deficiency in some of the beneficial bacteria in the colon. But at the other end of the spectrum, we see also an overgrowth pattern. So again, I flagged some of the potentially most clinically relevant organisms in this case. So there's a general overgrowth pattern, but we also see that Morganella is quite high at 1.69 E5, which is more than a couple orders of magnitude higher than the cutoff for being considered high. So again, that's pretty high. Also, once again, in this case, Pseudomonas was elevated, both the species Pseudomonas aeruginosa as well as other species in the Pseudomonas genus. Autoimmune triggers looked pretty normal, nothing notable there. In this case, no candida or viruses detected and no parasites detected. Once again, the intestinal health section, nothing was officially flagged as high or low, but we do see that the elastase is trending on the low end of normal. So just a side note is that we consider optimal for elastase on GI map to be around 500 and above. So this is well below that optimal range and would be considered suboptimal or low normal. Could be contributing to general kind of digestive dysfunction type scenario, which then may play a role in promoting dysbiosis. And there are studies that support the fact that pancreatic insufficiency or low elastase as a marker for pancreatic enzyme production may be related to microbiome dysbiosis and may in fact be a major factor in dysbiosis. So the case summary in this scenario is, once again, there's the low normal suboptimal elastase, general overgrowth pattern. High Morganella was also noted in this case. That's a possible contributor to histamine issues because Morganella is well established as a potential producer of high amounts of histamine. And then once again, we already talked about Pseudomonas, that could lead to possible issues with gluten and maybe the broader picture of food sensitivities, which of course may be related to the fact that this patient did have evidence of food sensitivities. So just a little bit more of the research behind some of this. This is a recent review article just published this year, Histamine Intolerance, Current State of the Art, where they talk about some of the key organisms that have been identified that can be especially abundant producers of histamine. And that would include Morganella as well as Klebsiella, which we also often do see elevated in patients that have common GI symptoms. Just kind of a side note is this is a figure from that article summarizing some of the key effects and things that are involved in histamine intolerance. So I've highlighted the area to the left that says, notes the symptoms pertaining to the gastrointestinal tract. So we can see at the top of that list is bloating, flatulence, postprandial fullness, diarrhea, abdominal pain, constipation, etc. So of course, those symptoms overlap quite a bit with what we commonly think of as symptoms of SIBO. So once again, there could be multiple factors happening in patients, whether or not they have SIBO. There may be essentially comorbidities or conditions that are also contributing to those same spectrum of symptoms as SIBO. So just want to take a side note here and emphasize that the small intestine, whenever there are issues in the small intestine, and that can be due to a wide range of factors. So we know, for example, that of course Pseudomonas, as I just talked about, may cause inflammation in the duodenum, the upper GI tract, other infections such as Giardia, cryptosporidiosis, and then also H. pylori infection, which sometimes can affect the duodenum, have all been shown to potentially cause some inflammation in the upper GI. That can have a negative impact on a couple of things. So often associated with more significant scenarios of inflammation in the small intestine is a reduction in the villi. So that results in a reduction in the surface area for absorption, but also may affect the brush border enzymes as well. So it turns out that brush border enzyme deficiencies are fairly common. That's becoming more and more recognized. And I'll have a little bit more to say about that momentarily. But just as a reminder, the key brush border enzymes that are in the upper small intestine include the disaccharidases. When there's a deficiency of those, that can lead to carbohydrate intolerance. The peptidases, when there's a deficiency of those may contribute to the production or the retention of allergens. So essentially those proteins can't be fully broken down, so they may retain their ability to be allergenic. Might also contribute to protein fermentation. So if proteins and peptides are not fully broken down, those can end up in the colon, where that can fuel a process called protein fermentation associated with dysbiosis also in the colon. There's also diamine oxidase, and so a deficiency there could lead to worsening histamine intolerance. So you can imagine if you have this combination of issues in the upper small intestine, say Pseudomonas is contributing to inflammation based on what we know from research, and then you have, in addition to Pseudomonas, also Morganella present in the GI tract that may be leading to higher production of histamine. Then you have a lowered ability to handle the histamine if the diamine oxidase is affected. So there can be a lot of consequences in this type of scenario. And if SIBO is an issue for the patient, SIBO itself may contribute to inflammation and other issues in the small intestine that may further compromise the function of these enzymes. Just a couple other things I'll note here is that intestinal alkaline phosphatase is another important brush border enzyme that actually detoxifies LPS, which is the pro-inflammatory molecule that is part of the outer cell wall of gram-negative bacteria. So that can be part of the picture as well, in particular with SIBO, because recent studies have noted that certain LPS-containing bacteria, such as E. coli and also Klebsiella, may be overgrown in SIBO. That may be characteristic of SIBO, at least for some patients. So once again, if there are issues compromising the function in the small intestine, then these brush border enzymes aren't going to be able to detoxify LPS and essentially carry out these other functions, which can just make that scenario worse. All right, so this is just a excerpt from a recent article having to do with disaccharidases. And in this article, they say when small intestinal carbohydrate-dense chyme exceeds the disaccharidase glycolytic ability and enterocyte absorptive capacity, the maldigested oligosaccharides that pass to the colon become subject to excessive fermentation. Resultant clinical features include abdominal distension to luminal retention of water and gases. So yet another potential contributor to these common symptoms. And as I'll get to if we have time towards the end, some researchers suspect that at least in some cases, that positive breath tests for some patients might actually be due to rapid transit that's basically reflecting increased colon fermentation. So not necessarily always is there a scenario where there's increased fermentation in the small intestine. In some cases, they may just have very rapid transit, and that may promote fermentation in the colon. This is a slide that summarizes a number of the potential causes of intestinal villus atrophy. So that's kind of the more significant, more extreme end of dysfunction in the small intestine. There's now a recognition that even conditions that may not lead to outright villus atrophy may still have negative impacts on small intestinal functions, such as low-grade inflammation. But this gives you a really good picture that there's many different things that can affect the function of the small intestine and then may have these negative impacts, particularly on the brush border enzymes and also on just general absorption and digestion. And I highlighted the area on the left that calls out some of the known infections that have been associated with villus atrophy, including some of the better known ones such as Giardia, Cryptosporidia, but also H. pylori, which is not necessarily as well recognized or appreciated, but in some cases, when it's affecting or inflaming the duodenum, that can lead to villus atrophy as well. All right, so now back to the third case. So this is a 40-year-old male. Breath test was positive in this case with high hydrogen and moderate methane. This patient was treated with rifaximin as well as herbal antimicrobial treatment and then some level of motility management in an attempt to control the SIBO symptoms. Post-treatment, a retest was done, breath test was done again, hydrogen was down, and then the CH4, the methane was only improved a little bit, which kind of went along with symptoms, that symptoms were somewhat better, but this patient wasn't quite at 100%. So once again, a pretty common scenario where treatment does seem to help, but there are still some lingering symptoms. In this case, pathogens were clear. Parasitic pathogens, viral pathogens were also negative. H. pylori was detected at a moderate level. Just a note here, so we do have the cutoff of 1.0 E3 as sort of the official cutoff for considering H. pylori to be at a high level. But we do sometimes see that patients may be symptomatic from H. pylori when it's only at the E2 level. And that's really where clinical judgment plays a big role because it is a bit of a gray area. And that's certainly true for many pathogens and opportunists that if they're just below the cutoff, that doesn't necessarily always mean that that's not clinically significant. So you may want to pay attention to these numbers, even if they're not flagged as high, and then assess whether it seems as to whether the symptoms and your overall assessment of the patient indicates that they might still have an issue. In those cases, you may want to consider addressing the H. pylori. In the normal bacterial flora section, we see quite a bit going on. Overall, there's an overgrowth pattern with the high Enterococcus, high Clostridia, high Enterobacter, and then under the phyla microbiota section, high Firmicutes. And then along with that, we also see there's a lack of Akkermansia, less than detection limit, and then a very low level of the Faecalibacterium. Those are two important keystone species indicative of the microbiome in the colon. And these particularly important species are obviously deficient here, indicating that there's a potential deficiency overall in the microbiome in the colon, along with an overgrowth pattern. Now, this is kind of a more complicated topic, but we do see pretty frequently that Firmicutes is one of the key markers that's elevated in patients that have these common GI symptoms. When you look deep into the research on factors that influence the overall level of Firmicutes, it appears that when there's an increase in influx of nutrients into the colon, particularly that first part of the colon, which is kind of the primary area for fermentation in the colon, especially carbohydrate fermentation, that can be due to either high carbohydrate diet, for example, or just consuming overall too many calories. So that can be one reason why it may be linked to the Firmicutes bacteroidetes ratio that's associated with obesity. But also it's been recognized that just poor digestion, where these carbohydrates may not be fully digested and absorbed in the small intestine, then can lead to an overgrowth of Firmicutes. So we often do see an overgrowth pattern that may be more indicative of overgrowth in the large intestine versus the small intestine that can still lead to common symptoms. In the opportunistic bacteria section for this patient, once again, we see that there's overall a high level of opportunists. Once again, kind of just the standard overgrowth pattern. Once again, Pseudomonas is one of the key organisms that we do see overgrown, along with a few others. So we already talked about Pseudomonas. So I want to talk about some of these others here. So I've also noted Enterococcus, as well as Staphylococcus and Streptococcus. So just want to talk about a little bit more of the research behind this when we get through the rest of this case. Potential autoimmune triggers section once again looked clear. Only the two markers that are normally present were detected. A little bit of Candida was also detected here. That's again kind of in this low level range that may or may not be clinically significant. That's really where the assessment of symptoms come into play. We often do see Candida present, whether it's a low level or officially high, in patients that have evidence of poor digestive function. So there may be a connection there between poor digestion and a higher likelihood of seeing Candida overgrowth. Parasite section, once again, was negative. In this case, when we look at the intestinal health section, we do see a couple things that were flagged as high. So there's the anti-gliadin, again, a marker for reactivity to gluten. That was at 200, so quite a bit above the cutoff for being considered high, indicating that there is some gluten sensitivity is the most likely scenario, although it's also possible that some patients may have additional reactivity to gluten cross-reactive foods. We do see here in this case that calprotectin was a little bit elevated beyond the cutoff, and so that's indicating that there's some level of inflammation in the colon in particular. So case summary, just summarizing these findings, is there was a moderate level of H. pylori. In terms of the normal bacteria, there was an overgrowth pattern along with low Akkermansia and Faecalibacterium, so kind of a typical dysbiosis type pattern. In the opportunistic bacteria section, there was an overgrowth pattern along with high Pseudomonas. And then Candida was positive at a low level. And then we also saw the anti-gliadin was elevated, high calprotectin was present, and also that elastase was suboptimal, which I didn't note, but if we go back there, so elastase is at 363. Again, optimal is considered to be around 500 and above. So this is really kind of in that middle ground where it may or may not be clinically significant. Some patients may benefit from digestive support if this is significantly suboptimal for them, but it's really not in a range that we would consider insufficient in any way. But again, it's sort of a suboptimal type scenario that may be contributing to the overall picture of reduced digestion. So one thing I want to just note here before we kind of dive into some of the studies again is we did see that Pseudomonas was elevated, and there is research, as I noted, linking higher Pseudomonas levels in the gut, particularly to food sensitivities and especially to gluten reactivity. And we did see in this case that the anti-gliadin was elevated. So that does suggest that this may be part of the symptomatic picture. This patient may have significant gluten sensitivity, or possibly, once again, sensitivity to those gluten cross-reactive foods. This is a reminder that H. pylori, there's a number of studies that have associated H. pylori with an increase in the incidence of hypochlorhydria. Now, I just want to point out that certainly not all patients that have H. pylori overgrowth necessarily have hypochlorhydria. There's sort of a spectrum where some patients may actually have high stomach acid, and that's thought to be dependent on exactly where in the stomach the H. pylori infection is occurring, such as antrum versus corpus. But in general, in chronically infected patients long-term, it's thought that that chronic inflammation can have a negative effect on parietal cells, that can lead to chronic hypochlorhydria for many patients. I'll skip over this because it's just getting into some of those details from that same paper. What I want to talk about here, though, is kind of this link between low stomach acid and this type of overgrowth pattern that we commonly see. So this is an article titled Proton Pump Inhibitors Alter the Composition of the Gut Microbiota. And this is an area that's been really well studied. There are quite a few studies that have been conducted looking at the effects of proton pump inhibitors on the gut microbiome. So we do see in research a very consistent pattern that translates quite well to what we see on GI map, not only when patients are on PPIs, but also other scenarios that can lead to low stomach acid, such as H. pylori overgrowth. So they say here we identified a significantly lower abundance in gut commensals and lower microbial diversity in PPI users, with an associated significant increase in the abundance of oral and upper GI tract commensals. In particular, significant increases were observed in Streptococcacea, which is the taxonomic family that Streptococcus is in. This is an example of another review article. So this is titled Intestinal Dysbiosis Secondary to Proton Pump Inhibitor Use. PPI use significantly increased the presence, once again, of Streptococcacea and also Enterococcacea, which are risk factors for C. diff infection, and decrease that of Faecalibacterium, a commensal anti-inflammatory microorganism. So again, we do see this pattern quite a bit, particularly with Streptococcus. So that's the one that's been noted most frequently in these studies as being linked to low stomach acid. So that's certainly one of the ones that seems to be most closely aligned. Can't really be considered, of course, a diagnostic marker for hypochlorhydria, but is apparently highly associated based on not only what we know from research, but also what we see clinically. And then some of the other organisms that are often elevated would include Enterococcus species and then Staphylococcus. There's also some studies that show that that can be increased with low stomach acid as well. And some of these others that we commonly see elevated, for example, when H. pylori is high, or when patients have evidence of not digesting well, there's a lot of overlap in those patterns. So there may be others that are also elevated when stomach acid is too low. So this is just the full abstract related to that previous one. So I just want to now dive into the fourth case example before we talk about a few other areas of research related to SIBO symptoms and how that can apply to GI map. So case example number 4, this is a report for a 50-year-old female who was diagnosed with SIBO with constipation. Did not appear to have elevated methane though, but did have constipation. 5 rounds of rifaximin plus multiple rounds of botanicals were done over the course of time, and that's in response to the fact that this patient does respond to the treatment, but then symptoms always return. So this treatment was just repeated again and again, and then they switched tracks to the botanicals to see if that produced better results. And unfortunately, this patient just kept repeatedly relapsing. So again, kind of the worst case scenario when it comes to SIBO, where treatment really doesn't have a long-term effect. In this case, we saw kind of an interesting pattern. So there was some C. diff overgrowth, and this is based on the detection of the toxin genes, in this case, indicating that the potentially pathogenic strains were in fact present and overgrown. Now, it's important to note that a lot of times patients can be carriers of these pathogens, and at least in the case of C. diff, that can often be due to the fact that they're not necessarily expressing their toxins. So again, we're looking at the toxin genes, not at the production of the toxin proteins themselves. So this is just confirming that the pathogenic strains are present. And of course, then making sure patients, assessing whether patients have the related C. diff infection symptoms or not is very important. In this case, there didn't appear to be any obvious symptoms related to C. diff. So it is there, but not necessarily causing significant symptoms in this case. And that's the same with this enterohemorrhagic E. coli, which we often do see present and sometimes elevated in patients that don't have the typical diarrhea or gastroenteritis type symptoms. So once again, they may just be carriers, but this certainly can reflect and be part of a dysbiosis pattern that may be due to certain physiological imbalances. So we did see earlier mentioned in one of those PPI papers that proton pump inhibitors can be one risk factor for not only C. diff, but also other pathogens. And that may be due to the fact that stomach acid is one potential barrier to various pathogens. So when it's insufficient, then that can of course allow them to get through in greater numbers. For this particular patient, the parasitic pathogens, viral pathogens were negative. H. pylori was present at a very low level, only at the E1 level. So at that level, it's generally not likely to contribute to significant symptoms, but once again, on a subset of patients, there may still be an effect. And typically, again, that has to do with low stomach acid. So the lower the level is, of course, the less likely clinicians are to want to treat that because that's not really considered significantly elevated, but it is present. So that may still have a minor impact for some patients. Under the normal bacterial flora, we see that the Bacteroidetes phylum was quite high at the E12 level. And then there were a couple of the commensals that were low, Akkermansia and then Enterobacter. So one thing I'll kind of point out as we get to the end of this report is some of the commonalities among the things that were elevated in this particular patient's report that's consistent with the potential for poor digestion. So we can see here under the opportunistic bacteria that two of them were elevated. Bacillus, that's something that we commonly see elevated in patients that have digestive issues. It's not really considered in any way pathogenic. There are a few pathogenic strains, but they're not common. So by and large, it's essentially kind of a normal bacteria group that's often overgrown as part of this digestive dysfunction pattern. And it is part of that same group that some of these others are, such as Streptococcus, that tend to overgrow as well. So in this case, I noted Streptococcus as being potentially clinically significant in the sense that that's a fairly high level when it's at the E4 range. And once again, it's often associated with low stomach acid. In this case, under the autoimmune triggers section, we can see that Fusobacterium was elevated just a little bit above that cutoff level of 1 times 10 to the power of 8, or 1E8. That one is known to be potentially inflammatory, but is also actually a common resident in the upper GI tract and the oral microbiome. And so in those scenarios, it's considered to be a fairly normal bacterium, not necessarily contributing to any particular issues, particularly when it's in the upper GI tract. Is associated though with inflammatory conditions when it's in the lower GI tract. So elevated levels sometimes can be associated with inflammatory conditions and also sometimes elevated calprotectin. No fungi or yeast were noted, also no viruses and no parasites. In the intestinal health section, we do see that the elastase was quite low, indicating insufficiency of pancreatic enzyme production, which may help to explain a lot of what we're seeing in this case with the various types of organisms that were overgrown. So of course, in this case, this is implying that digestion is not optimal, allowing these undigested foods to potentially promote the growth of certain types of bacteria that can take advantage of those foods. Now, when they get into the colon, there's a lot of research showing that excess protein, and particularly the breakdown products, amino acids, in the colon can promote the growth of certain opportunistic and pathogenic bacteria. So if we summarize the case here, C. diff plus the E. coli pathogen plus the high Bacteroidetes, those have all been linked to either higher protein diets or an increase in amino acids in the colon, particularly for the pathogens. So that's been shown in a lot of research in recent years. We don't have time to go into that detail today, but there's a number of studies published on certain amino acids, particularly proline, and proline-containing foods may promote the growth of C. diff. And for E. coli, in general, protein has also been shown to not only increase its growth, but also potentially increase its virulence for particularly the pathogenic strains. And then high Bacteroidetes, so that phylum has been linked to being elevated in cases where patients are consuming a higher protein, especially animal protein diet. We did see high streptococcus, again, that may indicate low stomach acid. We do know that low stomach acid, of course, is especially important for protein digestion. And part of that reason is due to the conversion of pepsinogen to pepsin in the stomach, which requires a sufficiently low pH in order for that to be efficiently converted. So low stomach acid may actually be related to more protein and more amino acids getting into the colon, which can then fuel the growth of these various pathogens and opportunists. We also noted high fusobacterium in this case. That's another example of an organism that thrives on amino acids. So it's considered asaccharidic, so it doesn't break down sugars and does tend to thrive more on amino acids and proteins. Low elastase would certainly contribute to that scenario. So if we have a combination of low stomach acid plus reduced pancreatic enzymes, that can certainly lead to reduced digestion, which may help explain why these certain types of microbes are overgrown in this case. Lastly, there was also low normal or suboptimal secretory IgA, which is something we often note that can be present along with lack of certain key beneficial bacteria. And so often it can be considered a marker for lack of key bacteria, and that can have some negative consequences as well. All right, so those are the 4 cases I'm going to cover. I do want to add one more thing here. So this is just showing you a couple of screenshots because we haven't talked much yet about the methanogens that are present on GI map. So I'm not going to go through a whole example here, but this is a screenshot of the opportunistic bacteria section. And you can see here that the Methanobacteriaceae family, which is in the opportunistic section, was elevated in this patient. So it's just above the limit here, but we don't see E9 levels very often in most patients. So there's actually quite a bit to know about the methanogens. I mean, the obvious thing is, of course, is that they potentially produce methane. When they're overgrown, they may produce it at higher levels. And that is something that we often see associated with patients that have constipation, which is certainly what we'd expect when they're elevated. But to understand how they kind of fit into the overall picture, we want to look at that a bit more. And I'll show you some research related to that momentarily, but oftentimes in these patients, we will see a general overgrowth pattern along with the high methanogens. And if you see here this is for the same patient, this is the normal bacterial flora section. Under the phylomicrobiota section, we can see that both the Bacteroidetes phylum and the Firmicutes phylum are elevated. So it appears that when both of these are elevated, that may reflect increased fermentation activity in the colon, which then can feed into promoting the growth of methanogens. So that is a scenario that we often see, and again, may be related in some cases to SIBO, or at least methanogen overgrowth, which is now being referred to as intestinal methanogen overgrowth, or IMO. If we look at this figure from a recent review paper, this is kind of summarizing some of the key dynamics in the colon that can lead to higher production of both methanogens as well as the sulfur-reducing bacteria, which produce hydrogen sulfide. So basically, to explain this figure, if you look at the bottom here, it says proximal colon on the left, and then distal colon on the right. So it's showing you kind of how the scenario changes as you go from left to right, basically from the proximal colon, the first part of the colon, to the distal colon. You can see on the left column, here it says growth, relative abundance. So it's basically kind of generally indicating the relative abundance of the different types of bacteria and some of the factors involved. So you can see here in the middle, on that left side in red is the Firmicutes phylum. They tend to be the ones that grow in the first part of the colon. So that's really where fermentation kicks in of carbohydrates. So they're especially geared towards fermenting those fibers and other types of non-fermentable or non-digestible carbohydrates, fermentable carbohydrates. And in that process, they break down hydrogen, or they produce hydrogen in the breakdown process, which can lead to butyrate, of course, which is a beneficial product. And then hydrogen, if it accumulates, may contribute to symptoms. But the role of the methanogens in this case is to consume that hydrogen and then produce methane. So in that process, it's helping to facilitate fermentation. So it's actually thought to be a beneficial process up to a point because it's facilitating this process that's resulting in production of butyrate. But of course, if that's excessive, if there's too much fermentation, such as from poor digestion, consuming too many carbohydrates, et cetera, or rapid transit, all of those things can lead to an increase in methane production. There's some details we can't really get into here, but depending on sort of the change in conditions and also just further along the GI tract, there can be a transition to more of the hydrogen sulfide type producing bacteria. And that's particularly where the Bacteroidetes also thrive, which tend to produce propionate. So towards the end of the colon, there's a switch from predominant butyrate production to more propionate production, kind of in a typical situation. And that's also where these proteobacteria, you can see those below here, the Bacteroidetes, those are also more predominant in this last part of the colon. And that's really where this whole scenario of protein fermentation can come into play. So bacteria generally preferentially ferment the carbohydrates. Once that is exhausted, then some of the species that are specialized in fermenting proteins, that's really where they come into play. And so that promotes the growth of Bacteroidetes. It can promote the growth of some of the proteobacteria as well. And then again, another byproduct of that, breaking down the sulfur-containing amino acids in particular, would be hydrogen sulfide gas. So this is pretty well worked out in research, and this is a great representation of some of those dynamics for understanding some of these, where the origin of some of these gases and then some of the factors that come into play. And this is the reference for that figure. So the article is titled Integrated Multidisciplinary Perspective for Addressing Challenges in the Human Gut Microbiome. And just want to read a brief excerpt from this article. So this is, again, from this article that figure is taken from, and they say an integrated perspective of the human gut microbiome and their linkages to human health will pave the way forward for delivering against the objectives of precision medicine, which is targeted to specific individuals and addresses the issues and mechanisms in situ, which basically means per patient or in the body. So in this case, we're really emphasizing how we can take all this microbiome information that's been coming out over the last few years and not just necessarily look at species individually and what do we do to address that particular species, but looking at it more as an integrative whole and how does that ecosystem work based on what we know so that we have a better understanding of how to approach that from a clinical therapeutic standpoint. All right, so that brings me back to kind of where we started, which is where clinicians often start to think of SIBO and deciding whether or not they want to recommend a breath test to test for whether SIBO is actually a problem for a patient, really based on symptoms. But I think you can start to see here that there's quite a few things that can lead to similar symptoms. There's a lot of symptom overlap in these various type of conditions and also related to various organisms. So of course, bloating, distension is common, excessive gas, abdominal discomfort, diarrhea or constipation. And that's really where clinicians start to think of things like SIBO. But what we're learning from not only research, of course, but also just the experience with GI map is that there can be many other things that also come into play. So if they may be coming into play alongside SIBO, or they may actually be the primary cause of symptoms and not SIBO in some cases. That's really underscored in this table from a recent review article. This was actually more related to IBD, but it's very relevant because it basically is talking about sort of differential diagnosis of symptoms and other things that can contribute to the symptoms that are often seen in IBD. And those overlapped to some extent with IBS and SIBO. So you'll note here on the left, there's a column for disease, and then also a column for symptoms. So starting with, I'll just go through a few of these. You can see bile acid malabsorption. Symptoms are commonly diarrhea or urgency, exocrine pancreatic insufficiency. Symptoms are often abdominal discomfort, bloating, diarrhea, or greasy stool. Carbohydrate intolerance, abdominal discomfort, bloating, diarrhea. SIBO, abdominal discomfort, bloating, constipation, diarrhea, distension, etc. Small intestinal fungal overgrowth, abdominal discomfort, bloating, diarrhea, distension, urgency, etc. So you can see there's a lot of different types of scenarios and conditions that can all lead to very similar symptoms that are overlapping, particularly when it comes to abdominal discomfort, bloating, and then alterations in bile or in bowel habits, such as diarrhea in particular. So it's really important to think of these other alternatives when you're thinking about what approach do you want to take for testing and assessment of the patient. All right, so I'm going to go ahead, due to lack of time, I'm going to go ahead and skip this one here real quick. So just to, again, this is reinforcing that these various conditions can lead to similar symptoms. So just be thinking of these things. And on GI map, we often do see evidence related to, for example, IBD, such as overgrowth of inflammatory type of bacteria, calprotectin is elevated, celiac, and certainly gluten sensitivity, candida overgrowth, hyperchlorhydria. We certainly get a good sense of that with GI map, and then pancreatic function. So these are some of the key additional factors to consider. So that's really brings me to the bottom line is common upper GI symptoms may not be due to just SIBO. They could be due to a wide variety of other imbalances. Just want to touch on this as kind of a last topic before we go to questions. So this is a recent review paper that I would certainly encourage everyone to check out if you haven't already. So it gets into some of the challenges with SIBO, both in terms of testing in particular and treatment. But this covers some of the controversies in SIBO. So some clinicians may not be aware of some of these issues. And so it's really good to be aware of these when you're selecting a test, deciding whether or not to test, et cetera. So to summarize some of those issues that are highlighted in that article without going through everything, typically there's issues related to the choice of tests. So the most common ones tend to be the glucose or lactulose breath test. Those can come up with somewhat different results, not only because they're different substrates, but they can reflect different parts of the small intestine. So glucose tends to mostly detect potential overgrowth in the upper small intestine versus lactulose, which may be able to cover more of the extent of the small intestine. Then there's also issues with the gold standard. So for many years, the gold standard has been considered to be culture from small intestinal aspirates, but even that has changed over the years. So it used to be that was done more in the jejunum, but now for sort of technical reasons, it's more common to assess the duodenum, which may have a somewhat different number of bacteria and also makeup of bacteria. Also, recent studies have looked at molecular testing, so essentially sequencing, DNA-based type testing, where they're able to look at a much broader range of bacteria and have found that the total amount of bacteria is sometimes up to 2 orders of magnitude higher than what's picked up by culture. So that really shows that the gold standard really is insufficient in that particular respect, that we're really only looking at the small fraction of bacteria in that case that can be cultured. Then there's also issues regarding the role of transit and also malabsorption. So there's some studies that indicate the early peak on SIBO tests in some cases may not actually be taking place or due to overgrowth or fermentation taking place in the small intestine, but may actually reflect large intestine due to rapid transit. Of course, then there's the debates over interpretation of results in terms of single peak, double peak, timing, et cetera. So there certainly are established guidelines now to help with those interpretations, et cetera. But of course, those of us who have looked at actual SIBO tests, they don't always follow that expected pattern. Sometimes it can be quite challenging to know what to make of it. I'm just going to highlight a couple of these real quick here and then start to take questions. So there's just one example of some of the several studies that have come out in recent years that have called into question some of the assumptions about SIBO and SIBO testing. This one is titled Small Intestinal Microbial Dysbiosis, Underlies Symptoms Associated with Functional Gastrointestinal Disorders. And they say, here we show that SIBO based on duodenal aspirate culture reflects an overgrowth of anaerobes, does not correspond with patient symptoms, and may be a result of dietary preferences. Small intestinal microbial composition, on the other hand, is significantly altered in symptomatic patients that does not correspond with aspirate cultures. So this is a bit of a controversial result. There's certainly been a lot of criticism of this study, a lot of discussion and debate over these types of sort of contrary studies that call some of the aspects of SIBO and SIBO testing into question. But it does really emphasize that newer technologies are helping us understand better what's going on in the small intestine, and particularly which particular microbes may be associated with symptoms in a given patient. And that's really where the precision medicine aspect comes into play. So once again, it's something to consider when you're considering different types of testing to assess patients with these common symptoms. I won't go through these others, but there are a number of other studies. You'll have the video recording, so you can basically go ahead and look at the recording to see these other references. But due to lack of time, I'm going to go ahead and skip them. All right, so really that kind of brings me to the final point, which is the role of advanced stool testing with GI map in this type of scenario. So when you're considering patients that present with these common GI symptoms and realizing that there can be many different things contributing to those symptoms, what do you start with as far as the test? You go straight to, if you suspect SIBO, for example, do you go straight to breath testing? Is it better to do a comprehensive stool test, et cetera? So some things to keep in mind when it comes to comprehensive stool testing is that it really helps provide an overview of the gut ecology and physiology. Unlike a breath test, which is really just telling you one specific thing, whether there's excessive fermentation in response to that particular sugar that was studied within a given time, which may reflect SIBO, but then again, there are these studies that call that into question, that it's not entirely clear in some cases if that's not due to something else. Also, comprehensive stool testing can help you identify possible common causes of symptoms that are SIBO-like or IBS-like, such as H. pylori overgrowth that can lead to hyperchlorhydria, candida, et cetera, which is that next bullet point, identify multiple factors that may be contributing. Sometimes you may have more than one factor. So you may have a combination of hypochlorhydria, poor pancreatic function, so reduced digestion, potentially malabsorption as well. And then also overgrowth of certain opportunists. In some cases, those can further contribute to symptoms such as candida and pseudomonas. So really getting into the details is one of the key advantages of comprehensive stool testing for patients that have these common symptoms. And in particular, if SIBO does really appear to be the case, and if SIBO breath test does confirm that, but then patients are not responsive to the treatment, a comprehensive stool test can be a great way to help you understand what other things may be going on with that patient, other factors that you may want to consider addressing. All right, well, hopefully that helps illustrate some of the key challenges that are often faced with SIBO and SIBO testing, and then how GI-MAP can help shed some light on some of those potential factors that may explain SIBO-like symptoms or may help explain why patients are not responding well to treatment.